Happy morning, children. This is chapter 13, unit 5, plant physiology. In that functional organization, we are going to deal with photosynthesis. It's a very important and interesting topic. So here in this topic, we'll be dealing about the historical events, definitions, significance, site of photosynthesis, photosynthetic pigments, and spectrum of electromagnetic radiation, photosynthetic unit, absorption spectrum and action spectrum, immersion experiment and ill reaction, and then you have the reaction. Okay, that is a modern concept of photosynthesis, phases of light, phases of photo-oxidation phase, photochemical phase, photophosphorylation, chemosmotic theory, dark reaction, H&Slack pathway, CAM cycle, respiratory, photorespiratory, factors affecting and photosynthesis in bacteria. Let me divide this lesson into 4 parts. Okay, so the beginning, the historical evidence, significance, site of photosynthesis, photosynthetic pigment, the radiations, photosynthetic unit, and all this till 13.7 will take it as a first session. The second section, it will be from concepts, photo-oxidative phase and photochemical phase with photophosphorylation and chemosmotic theory. So till this, we'll take it as a second reactions. And the third one will be dark. Okay, this particular thing, the third part. And the fourth part will be dealing with the C2, C4 cycles, the different types of photosynthesis taking place in plants. And the last session would be factors affecting and photosynthesis in bacteria. So this is a bit bigger lesson, but let's see the content of the things that is going to be dealt in this lesson. So the first historical things are given over here: Von Helmont, Stephen Hales, Joseph Priestley. So I want all of you to read this. It would be fine if you prepare a tabular column. Children, for doing your entrance, surely you will get answers, questions from this. So you prepare a tabular column making the name of the scientist, the year, and what they contributed. So hardly you have not more than 20 people here. So go with these names, you prepare a chart and you learn what they have contributed for photosynthesis. The next one, definition. Definition, right from your smaller classes, lower classes, you would have learned photosynthesis means preparing of food by the process of using sunlight. But here it is a different definition. Refer to photochemical oxidation and reduction reaction. Okay, I repeat again, photochemical oxidation and reduction reaction with the help of light, converting solar energy into chemical energy. Okay, sunlight is there, the sunlight is converted to chemical, that is the food. So this would be your definition for photosynthesis. So photosynthesis is photochemical oxidation and reduction reaction carried out with the help of light, converting solar energy into, this is the definition which you will have to write in your exam if at all it is asked. For this, you need a certain raw materials. The raw materials like carbon dioxide, okay, carbon dioxide, water, and with the help of sunlight. Okay, so you need carbon dioxide, which should be the first thing, then the water and the light energy. Along with this, you also need one more thing, that is the chlorophyll. It is given out in an overall equation, that is CO2 plus H2O. CO2 is carbon dioxide plus water, H2O. Okay, in the presence of sunlight and chlorophyll pigment gives out C6H12O6. This is nothing but carbohydrate. C6H12O6 is the food molecule, is the energy substance, it is a carbohydrate, and it gives out oxygen. As we need 6 carbon here for carbohydrate, we are equating the equation now. So 6 carbon dioxide plus 6 H2O in the presence of sunlight and chlorophyll pigments get converted to carbohydrate plus 6 molecules of oxygen which is liberated out. Rubin and Kemen is demonstrating it in a different way. 6 molecules of water as insufficient for the evolution of 6 molecules of oxygen. So it is equated as C is taking double the amount of water. Okay, when you take double the amount of water, you have water also liberated here. So it is a collection of oxidation and reduction reaction, which we call it as the redox reaction. Oxidation, water is oxidized into oxygen with the loss of water, and it is called reduction because carbon dioxide is reduced into carbohydrate by gain of electron. So two reactions are taking place. One, oxidation is also taking place by losing of electron, and reduction is taking place by gaining of electron. Therefore it is called as, what is it called as? Redox reaction. Therefore it is called as redox reaction. So photosynthesis is also known as the redox reaction. Why? You have to explain this for it. And now the significance. What are the importance of photosynthesis? It provides food for all living organisms, and it balances the oxygen level, and it balances the oxygen and carbon cycle in the nature. It is a main source of fuels. Okay, coal, petroleum, it provides fuel, and it is a primary producer, and it provides fodder, fiber, firewood, timber, useful medicinal products. So these are the various significance of the photosynthesis. Where does this photosynthesis takes place? It takes place in chloroplast. Okay, it takes place in chloroplast. Let me introduce one more thing. Photosynthesis is not a simple process. It is of two steps. One is light reaction, the other one is dark reaction. So photosynthesis is done by two reactions. Keep in your mind light reaction and dark reaction. For doing both this reaction, we need an organelle, which is the chloroplast. Let's learn what is a chloroplast. Okay, so the chloroplast is a double membrane bond structure. Outer membrane, it is a discoid shape. Okay, discoid means disc-like structure. It has got two membranes. This is outer membrane and the inner membrane. This membrane is completely filled with stroma. It is filled with stroma. This is outer membrane and this is inner membrane. Okay, and this stroma is also known as matrix. It is also known as matrix. And they consist of thylakoid. Thylakoid is a coin-like structure. And this thylakoid, when it is arranged one above the another like piles of coin, 5 ரூபா coin ஒன்று மேல அடுக்கி வைக்கிறோம். The single one is called the thylakoid. Single one is called thylakoid. When it is arranged one above the other, it is called the grana or granum. In a chloroplast, you will have 40 to 80 granas. Okay, you will find granas everywhere. 40 to 80 granas will be present. And each grana is connected by a connection which we call it as frit. It is called as frit. So these are the basic structure. I have drawn only one. Outer membrane, inner membrane, matrix or stroma, single unit called the thylakoid. Thylakoid, when arranged one above the another, it is called grana. And in a chloroplast, you will have more than 40 to 80 granas. And each grana is connected with a connection which we call as laminar frit. Okay, and you will have ribosomes here and there. Ribosomes will be there, and you will have DNA molecules. DNA will be there. Okay, so all these are present in the chloroplast. This chloroplast, the thylakoid in the chloroplast of grana is the region where the photosynthesis takes place. So photosynthesis, the site of photosynthesis is the thylakoid in the chloroplast. So this is a structure where the photosynthesis takes place. Now, for photosynthesis, we need pigment. The pigments are the chloroplast, chlorophyll pigments. Okay, types of photosynthetic pigments: chlorophyll, carotenoids, and phycobilin. In chlorophyll, you have got 9 types of chlorophyll. Chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll A, bacteriochlorophyll A, bacteriochlorophyll B, chlorobium chlorophyll 650, chlorobium chlorophyll 666. And here when you say carotenoids, you have keratins and xanthophylls, okay. And in phycobilin you got two, one that is phycocyanin and phycoerythrin. So these are the pigments that is necessary for photosynthesis. Of this, chlorophyll A is the primary pigment and it is the reaction center, and all other pigments are called the accessory pigments. So very important, chlorophyll A is the primary pigment and all the others are the accessory pigments. Now let's move to the important topic of light. The pigment chlorophyll structure is given over here. This is the structure of chlorophyll A and this is the structure of chlorophyll B. You can just look into it in your textbook. And now we'll move on to our spectrum of electroradiations. The rays at which the chlorophyll pigments will be absorbed, okay. The wavelength of solar radiation which reaches the earth is between 300 to 2060 nanometer, and the visible spectrum range is 390 to 763 nanometer. The color of light is determined by the wavelength, okay. So it is given over here, and you can find the electromagnetic consists of 8 types of radiations: cosmic rays, gamma rays, X-rays, UV rays, visible light spectrum, infrared, electric rays and radio rays. Of this, the visible light which is accepted by the reaction is very important. Now let's move to the photosynthetic unit called the quantasomes. What is quantasomes? Morphological expression of physiological photosynthetic unit, okay, is called the quantasomes, okay. Each quantasome measures about 180 Armstrong to 160 Armstrong and 1000 Armstrong thickness. So the unit of light that is taken by an organism which is called the quantasomes, and it is given over here. And this term was coined by Park and Begin, okay. Park and Begin coined the term quantasomes. And you will have the next topic, absorption spectrum and action spectrum. What is absorption spectrum? The term absorption refers to complete retention of light without reflection or transmission, complete absorption, okay, and it is called the absorption spectrum, okay. They don't reflect, they just take in whatever is given to them. It is chlorophyll A and chlorophyll B absorbs quantum from blue and red region, and maximum absorption peak is different form of chlorophyll A. Chlorophyll A 680 and chlorophyll A 70 functions as trap for PS2 and PS1 respectively. Now let's see action spectrum. The effectiveness of different wavelength of light of photosynthesis is measured by plotting against quantum yield. The curve showing the rate of photosynthesis is called action spectrum. Here, how the reaction taking place is called action spectrum. Absorption is just absorbing of the plant. You can see in this graph, the wavelength of light is given over here, and this is the photosynthetic rate. How does the photosynthesis takes place? And you can find 3 different lines over there: absorption spectrum and action spectrum at 3 different chlorophyll A and keratins and chlorophyll B. How do they do their reactions? Now let's move to the experiment given by Emerson, which is called as Emerson drop first effect, okay. What do they say is, at certain point the absorption will be more, okay. At certain level, when the light intensity goes down, it will be dropped out, okay. Emerson modified his experiment by supplying shorter wavelength of light. He found that the monochromatic light of longer wavelength, when supplemented with shorter wavelength of light, photosynthetic yield and required red dot. This enhancement of photosynthetic yield is referred as immersion enhancement effect. Before that, we should know what is this experiment. So they pass on the light. At certain point of the light, the photosynthetic reaction drops down. Therefore, it is called immersion drop effect. This thing, when changed with different types of wavelength, the